Hello everyone and welcome to video 11. So today we're moving on to key area 6, which is respiration. And to get the consumables and activities you need to go to 1 cell biology and then 6 respiration and then activities and consumables, and you can also get access to the PowerPoint. So the starter for today, because you should now be experts in genetic engineering. If you're struggling with genetic engineering, then you need to go back and look at the last video. So the diagram below represents some of the stages of genetic engineering which are used to produce medicines such as insulin for human use, and then you've got the diagram. Then it says complete the table below to identify the labelled parts of the diagram. So you've got to name the structure and you've got to put the letter beside it. So bacterial cell, what the letter is, insulin, gene, what the letter is, plasmid, what the letter is. Then for B, you've got to describe the next stage needed to produce insulin for the use as a medicine. And for C, you've got name another hormone produced by genetic engineering. So if you want to pause it and try and answer the questions. So the answers to the questions are: the bacterial cell would be E, your insulin gene would be C, and your plasmid would be D. And describing the next stage, so you've obviously got your desired gene, which was your insulin gene, identified, and then your genes being extracted, which you can see for C, and then your plasmid's being extracted, which you can see from going from E to plasmid, and then your plasmid's being cut open, and then your desired gene has been inserted, and then your plasmid containing your desired gene has been inserted to your bacterial cell. So you've been asked for the next stage to this, so that would be that your GM bacteria containing your plasmid and your desired gene would reproduce and multiply. And then after that stage, you'd have that your product would then be extracted and purified. So then it says name another hormone, like a human hormone produced by genetic engineering, and you should know two of these. So one's obviously said already, which is insulin. So the other one that you got told to remember was your human growth hormone. So if you managed to get all of them correct, well done. If you didn't, you need to go and look at the genetic engineering tutorial. So we're moving on to key area 6, respiration. So basically all of your toys that need So we're moving on to look at key area 6, respiration. So basically all of your toys that require electricity or anything that requires electricity will either have a main source of wires going to it, or it will usually have to have batteries. So basically our source of energy is what we're looking at today. So what makes us able to move, what makes us able to breathe, what is providing us with this source of energy. So your learning intention is to understand how energy is released from glucose and be able to describe it, and also understand what ATP can be used for and what ATP is. And for to be successful, I'm looking for you to be able to explain that the chemical energy stored in glucose must be released by all cells through a series of enzyme-controlled reactions called respiration, and also to be able to describe that the energy released from the breakdown of glucose is used to generate ATP, and ATP can then be used for cellular activities such as muscle cell contraction, cell division, protein synthesis, and transmission of nerve impulses. So respiration, what we're going to learn in this video tutorial is what it is, where does it occur, how does it occur, what are the substrates, so the beginning, and what are the products, so what is it given at the end, and why does it happen. So you need to make a note and you need to know this. So you need to know that respiration is the chemical energy stored in glucose must be released by a cell through a series of enzyme-controlled reactions called respiration. So you need to say this over and over again or write it over and over again. So in this diagram we've got our donut, which contains lots of glucose, and we have Homer Simpson eating our donut, and then this is allowing him to sleep, or it might allow him to exercise, etc. So the process in which sugars, glucose, are converted into usable energy, ATP, is what respiration is. The chemical energy stored in glucose must be released by cells through a series of enzyme-controlled reactions called respiration. So we know the law of conservation of energy. We should have learned this in second year. So energy cannot be lost or gained, it can only be changed from one form to another. So food is a form of chemical energy. So if we want to measure the chemical energy in food, we would need to change it to another form of energy. So it can be changed to heat energy, which we can then measure, whereas we can't really measure chemical energy. So you can measure heat energy by measuring the temperature. So there are three main food groups that we get. We get protein, fat, and we get carbohydrate. So to measure the energy content of food, we need to measure how much heat is given off by the food. So to do this, we would use a deflagrating spoon, which is this here, and we would put our little piece of food in it, and then we would burn it in oxygen or air, and we would see how much the temperature went up on the thermometer and how much the water above heated. So when a food is burned in air or oxygen, its chemical energy is converted to heat energy. The increase in the water temperature can be used as an indicator of energy content. So 4.2 kilojoules required to raise the temperature of 1000 grams of water by 1 degree Celsius. Now it's very unlikely that you get a question on this, and if you do get a question on this, you're usually provided with an equation. So I wouldn't worry about it too much. So we've got to try and answer the following questions. This is a bit of problem solving. So how is energy released in food? And name one problem with this experiment. So you can pause the slide and try and answer the questions. So the answers to these questions would be energy in food is released as heat energy, and heat energy would be lost to the surroundings as it's open. So it wouldn't all be going into the heating of the water. So another method to measure how much energy is released by food is to use a calorimeter. So as you can see, it's this contraption here. So you've got your food sample in your crucible, you've got your igniter, and then you've got your water surrounding it, and then you've got your filter pump, and then you've got your water, and then you've got your thermometer. So again, you would measure how much the temperature would change by the thermometer after you heated all your food, and it's asking why does the calorimeter give a much more accurate energy value than the other method, and that is because our heat can't be lost to our surroundings because the water is trapping it, so there'll be not as much heat lost to the surroundings. So energy transfer, so we've got our food, so like an apple for example, then we have to digest it, and then we have to get our energy somehow, and then we use that energy to move or to dance. So ATP is really, really important. It's important that you put it as capital A, capital T, and P because all of the different letters stand for something. So it actually stands for adenosine triphosphate. So energy is always released in the form of ATP, and think of ATP as a bit like a battery of stored energy. So this is the part that we are wanting to get to. So we're wanting to get our glucose to make ATP. So we want to get this ATP released from our glucose. So you would need to write a note on ATP, and it's very, very important. So think of ATP molecules as high energy compounds or batteries that store energy. Anytime you need energy to breathe, to tie your shoes, or to cycle 100 miles, your body uses ATP. So your body constantly needs to make new ATP as you use up your energy. So a human makes 90 quintillion ATP molecules per second, which is a huge, huge number. So you would want to make a note on adenosine triphosphate ATP. So the energy released from the breakdown of glucose during respiration is used to generate ATP. So it's an energy-rich storage compound, so it's your chemical energy. So this is made up from an adenosine molecule and 3 inorganic phosphates. That's why there is tri meaning 3, known as pi groups. So you've got your adenosine and you've got your free phosphate groups, and you want to write this down. So ATP is generated from ADP, which is adenosine diphosphate, di meaning two, and inorganic phosphate, so PI. So your adenosine diphosphate and PI come together to give you your adenosine triphosphate. So the energy stored in ATP is released when the bond which attaches the terminal phosphate is broken. So this bond here, terminal just meaning the end. So you need to write down a note on what ATP is used for, and it's a really important note because it comes up so often in exam questions. So you can title it the role of ATP. So the energy released from breaking that terminal phosphate of ATP can be used for cellular activities such as muscular contraction, cell division, protein synthesis, transmission of nerve impulses, active uptake of ions and molecules, which you know as active transport, and carbon fixation during photosynthesis. So if you get asked what can ATP be used for, you would say one of these. So it's important that you know this, so read it out a few times, write it down a few times, tell people about it. So the transfer of chemical energy by ATP. So this is just a summary of what happens. So you get your glucose and oxygen, and then that makes carbon dioxide and water, and that's coupled to the production of ATP. So this allows your breakdown of your glucose, allows your ADP and PI to join together to form ATP, and then your ATP can then be broken down to release energy, which gives the body energy for like muscle contraction or for protein synthesis. So this is an example of protein synthesis. So your amino acids can then build up to make your proteins. So you can write this and note this if you want. So now we're moving on to the next part. So we've looked at ATP, so we should now be experts on ATP. Remember, the most important note is to know what ATP is used for. So muscle contraction, nerve impulses, active transport, nerve transmission, carbon fixation, and photosynthesis. So now we're going to learn to describe glycolysis. So to be successful, you need to know that glycolysis is the first stage of respiration. You need to be able to identify that glycolysis takes place in the cytoplasm. You need to explain that glucose, the substrate of glycolysis, is broken down to two molecules of pyruvate, and you need to know that 2 ATP are produced at this stage. So the breakdown of glucose. So the first stage of respiration, glycolysis, and you need to know the name of the first stage, involves the breakdown of glucose by a series of enzyme-controlled steps to form two molecules of pyruvate. So during this process, two molecules of ATP are also produced, and these reactions take place in the cytoplasm. So you want to know, so put the breakdown of glucose as your title, and you need to say all of this. So you need to know that the first stage of respiration is called glycolysis. It involves the breakdown of glucose by a series of enzyme-controlled steps to form two pyruvate molecules, and then during this process, two molecules of ATP are produced, and these reactions all take place in the cytoplasm. Now, because it's controlled by enzymes, it'll only work at certain temperatures, because remember, if you increase the temperature, then you increase enzyme activity until you reach its optimum, which is usually around about 37 degrees, and then above that it'll denature because the enzyme's active site will change shape and it will no longer bind to the substrate. So this is a diagram, so if you learn better by visuals, this will be good. So your glucose is broken down to your two molecules of pyruvate, and at the same time your 2 ADP and PI form 2 ATP molecules. So glycolysis, breakdown of glucose into two molecules of pyruvate, 2 ATP formed, and that's what you need to know, and it happens in the cytoplasm. So after the first stage, glycolysis, you have these two molecules of pyruvate. So your fate of pyruvate depends on whether oxygen is present or absent. So basically your pyruvate can go either one of two pathways. So it can go with oxygen to the aerobic pathway, or it can go with no oxygen to the fermentation pathway. So firstly, we're going to look at what happens to pyruvate when there is oxygen. So we're going to look at the aerobic pathway first of all. So your learning intention for this part is to be able to describe aerobic respiration. And for to be successful, you need to know that aerobic respiration has to have oxygen to happen. You need to be able to identify that the second stage of aerobic respiration happens in the mitochondria, and you need to explain that pyruvate is further broken down in the mitochondria to carbon dioxide, water, and many ATP. So aerobic respiration is a process that happens when pyruvate has oxygen. When oxygen is present, aerobic respiration occurs inside the cell's mitochondria. Only when oxygen is present will the mitochondria let pyruvate in. If oxygen is present, pyruvate does not get to go in. It's not good enough on its own. So this is a mitochondria seen under an electron microscope. We've already had a look at the mitochondria, and that is why it is the site of aerobic respiration, which we learned in the first video. So your mitochondria has its outer membrane, it's got its inner membrane folds, which give it a large surface for respiratory processes. So its matrix contains enzymes. So cells like muscle, sperm, and neurons have many mitochondria, and that's because they have a high energy requirement. And we'll find out why that mitochondria means that it's good if a cell has a high energy requirement. So aerobic respiration. For aerobic respiration, it's important that you remember the saying, go cow. And we'll come to why that's important in a second. So when a molecule of pyruvate diffuses from the cytoplasm into the mitochondrion, it is broken down, resulting in the formation of water, carbon dioxide, and a large number of ATP. So you need to write that down and you need to know it. So you need to say it over and over again, write it down over and over again. Aerobic respiration is a hard subject to understand, so make sure you go over this over and over again. Lots of people get questions in respiration wrong. So an aerobic respiration summary. So glucose is broken down to two molecules of pyruvate, releasing enough energy to yield two molecules of ATP in the cytoplasm. If oxygen is present, each pyruvate molecule diffuses into the mitochondria, where they are broken down to carbon dioxide and water, releasing enough energy to yield a large number of ATP molecules. Now this equation at the bottom helps. So if there's glucose and there's oxygen, it gives you carbon dioxide. So carbon dioxide and water and many ATP. So to remember aerobic respiration, you need to remember go cow. So G standing for glucose, O standing for oxygen, reacts to form carbon dioxide, which is your CO part, and water and many ATP. Okay, sometimes you get asked about your substrate. So your substrate would be glucose in this case, and your products would be carbon dioxide, water, and many ATPs. Now sometimes you'll get asked what your high energy product is, and your high energy product is always your ATP. So you need to spend a lot of time on this little part of the video, so you might want to pause it and you might want to go over it again and again and again. So this shows you the whole of the summary of aerobic respiration in a diagram. So your first stage is called glycolysis and your substrate is glucose, and firstly it is broken down into two molecules of pyruvate and two ATPs are produced, and this happens in the cytoplasm. Then because oxygen is present, your pyruvate can then move into the mitochondria. So when your pyruvate is moved into the mitochondria, it can then be broken down into water and carbon dioxide, and at the same time it produces a large number of ATP which can be used. So if we remember rightly, our ATP can be used for several processes. So you can pause the video and write them down. So the things that ATP are used for are muscle contraction, protein synthesis, nerve transmission, well, the transmission of nerve impulses, active transport, and carbon fixation in photosynthesis. And to make this easier, we need to remember this saying, go cow. So if you've learned aerobic respiration properly, you should be able to answer these questions. So if you pause the video and answer the questions. So the diagram below shows stages in the breakdown of glucose in the presence of oxygen to form the final products gas X and substance Y. So you've got glucose to pyruvate with your ATP, and then pyruvate to a gas X and substance Y with your ATP. And it says identify gas X and substance Y. So gas X, substance Y, and then it says state the number of molecules of ATP which are produced per glucose molecule during each of the following pathways, so aerobic respiration and fermentation. You might not be able to get that one. And then C, state the location of the fermentation pathway in a cell. You might not be able to get that one either, but if you change the word of fermentation to glycolysis, which is the first stage, and you do that the same with the one above, and then you'll be able to answer the questions on fermentation after we go over it. So glycolysis and glycolysis. So for A, identify gas X and substance Y, the answer to that would be gas X would be carbon dioxide because goat cow, and substance Y would be water. So it then says state the number of molecules of ATP which are produced per glucose molecule during each of the following pathways. So aerobic respiration would be many ATP or a large number of ATP molecules. And for glycolysis, it would be 2 ATP molecules. And then state the location of the glycolysis pathway in the cell, that would be in the cytoplasm. So you should also be able to answer these questions. So one is, what is the substrate of respiration? Two is, where does the first stage of aerobic respiration take place and what is it called? Three is, how many ATPs are created at the end of aerobic respiration? And four is, what processes are ATP used in? So if you pause the video just now, and then I'll go over the answers. So the answers to this are, the substrate would be glucose, and then the first stage of your aerobic respiration happens in the cytoplasm, and it is called glycolysis. For 3, a large number of ATP molecules are produced at the end of aerobic respiration. That's why it is so handy for your high energy required cells to have lots of mitochondria, because it means there's more ATP produced because there is more aerobic respiration. So that's why nerve cells, sperm cells, muscle cells will have lots of mitochondria, and that is a common question that gets asked. And for 4, what is ATP used for? So muscle contraction, protein synthesis, active transport, cell division, transmission of nerve impulses, and carbon fixation during photosynthesis. So well done if you got all them correct. So now we are moving on to the other pathway. So we knew that after glycolysis, which is the first stage, are 2 molecules of pyruvate that were produced could go either of 2 pathways depending on whether oxygen was present or absent. So we looked at what happened when oxygen was present by the aerobic pathway, and now we're going to look at what happens when it's not present by the fermentation pathways. So we need to be able to describe fermentation, and that's your learning intention. And to be successful in this, you need to know that fermentation happens in the cytoplasm. You need to explain that in animals, pyruvate is broken down to lactate, and this can make muscles sore and fatigued, which just means tired, and this is reversible with oxygen. You need to explain that in plants and fungi, pyruvate is broken down to carbon dioxide and ethanol, and this is irreversible. So during fermentation, only 2 ATP are released, which is a lot less than what happens in the aerobic respiration. So fermentation pathway with no oxygen. So in the absence of oxygen, the breakdown of glucose begins normally, yielding 2 molecules of ATP. So however, the pathway resulting in the complete breakdown of pyruvate, carbon dioxide and water cannot take place, because without oxygen, the pyruvate cannot enter the mitochondria. Aerobic respiration happens in all cells the same, but fermentation is different in your different types of cells. So first we're going to look at fermentation in animal cells. So this can occur during vigorous exercise where muscles do not receive enough oxygen. So for example, if you were doing a sprint. So when animal cells respire without oxygen, they produce lactate. So this happens in the cytoplasm. So your glucose goes to pyruvate, which is your first stage, which is glycolysis, and your 2 ATP are made. And then your pyruvate is then further broken down into lactate. So lactate is the thing that, if it builds up, will make your muscles sore. So the buildup of lactate in the muscles reduces their efficiency, causing muscle fatigue and tiredness. So during this time, an oxygen debt builds up, and the oxygen debt is repaid when oxygen becomes available during a rest period. So after you do a really, really fast sprint, if you rest, then your oxygen debt will be paid back and you won't feel your muscles being sore and tired anymore. So the lactate produced can be converted back to pyruvate, and therefore this is a reversible process because both of them are liquids. So pyruvate can be converted to lactate, and lactate can be converted back to pyruvate. So here is a summary for fermentation in animal cells. So this is a diagram summary basically. So you've got your glucose going to your pyruvate, which occurs in the cytoplasm and is called glycolysis. So you've got your 2 ADP and 2 PI going to your 2 ATP. And then your pyruvate is then broken down to lactate when there's no oxygen. And then when oxygen is returned, your lactate can then be converted back to pyruvate. So you'd want to make a note on this and write it down. So fermentation in plant cells and yeast cells is different from what it is in animal cells. So this can occur in the root cells of plants when it's waterlogged soil, or in yeast cells when it's deprived of oxygen during beer and wine making. So this type of respiration is known as fermentation, and when plant and yeast cells respire without oxygen, they produce ethanol and carbon dioxide. So this is just a summary diagram of fermentation in plants and yeast cells. So we have the first stage, which is our glycolysis. So that happens in all of the different processes, and your glucose is broken down to 2 molecules of pyruvate, and then you get 2 ATP released. Now, because there's no oxygen, then your pyruvate is further broken down into ethanol, which is used in the production of alcohol, and also carbon dioxide, which can be used to make bread rise. So this process occurs in the cytoplasm and it's irreversible because this time it's a gas, so it can't be reversed. So glucose to your 2 molecules of pyruvate and 2 ATP is made. Then you get the production of ethanol and carbon dioxide. So you need to be able to compare aerobic respiration with fermentation in plants and with fermentation in animals. So you need to go probably back in this video and make sure you've got all 3 of the processes written down and you know them very, very well because it is very, very complicated. You can make up sayings to try to remember it. So to summarise the fermentation process and everything, so in glycolysis, the first stage, glucose is broken down to 2 molecules of pyruvate, releasing enough energy to yield 2 molecules of ATP. In the absence of oxygen, the fermentation pathway takes place. So in animal cells, the pyruvate molecules are converted to lactate, and in plant and yeast cells, they're converted to carbon dioxide and ethanol. All of these processes happen in the cytoplasm. So you need to make sure you know this, you need to read over it, you need to make a note of it, so write it out a few times, say it a few times, and make sure you know it. So to summarise both your aerobic respiration and fermentation and compare them both, you're going to do this next activity. So you need to draw out the table and you need to pause the video and try to answer the question. So where is the location of aerobic respiration and fermentation? Does it require oxygen? And how many ATPs are produced in aerobic respiration and how many in fermentation? And what are the end products in aerobic respiration and what are the end products in fermentation animal cells and then plant and yeast cells? So the answers are, so for the location of aerobic respiration, we should have got cytoplasm and mitochondria, and for the location of fermentation, it should be cytoplasm. For is oxygen required? It should be yes for aerobic respiration and no for fermentation. And the number of ATP produced per molecule of glucose should be large numbers of ATP for aerobic respiration and 2 for fermentation. And then your end products in aerobic respiration should be carbon dioxide, water, and many ATP. And for your end products in fermentation in animal cells, you should have got lactate and 2 ATP. And for your end products in fermentation in plant and yeast cells, you should have got ethanol, carbon dioxide, and 2 ATP. So very well done if you got that all correct. If you didn't get it all correct, then you should probably go back to the start of this video tutorial and look over aerobic respiration and fermentation again. So this question is to review your knowledge. So the list below contains some features of respiration in germinating peas. Now we know peas are plants. So the list says W does not require oxygen, X releases carbon dioxide, Y produces lots of ATP per glucose molecule, Z produces ethanol. So it says complete the table below by entering the letters from the list in the correct boxes to match the features with the type of respiration occurring. So each letter may be used more than once or once. So you've got your aerobic respiration in germinating peas and you've got your fermentation in germinating peas. So if you guys pause the video and you try to answer it on your own, and then I will give you the answers. So the answers are for aerobic respiration in germinating peas. Well, we know that it requires oxygen, so it's not going to be W. We do know that it releases carbon dioxide because go cow. We know it produces lots of ATP per molecule, per glucose molecule, but it does not produce ethanol. So the two answers for aerobic respiration are going to be X and Y. So for fermentation in germinating peas, we know it does not require oxygen, so it's going to be W. We know it releases carbon dioxide, so it's going to be X. It does not produce lots of ATP per glucose molecule, but it does produce ethanol, so it's going to be Z. So we should have X and Y for aerobic respiration, and we should have had W, Z, and X for your fermentation. So now that you have completed aerobic respiration and fermentation, you need to go review your knowledge. So if you go to the Bite Size website and you complete the revision and do the test till you get 100%, then it'll help you become more confident in everything. And I think there's a video there as well. And then if you can try and complete the How to Pass National 5 biology questions on page 34 and 35, and if you get stuck, you can email your teacher. So the next learning intention is to describe what respirometers are. And for you to be successful, you need to be able to explain how to use a respirometer. You need to be able to practice problem solving questions and know how to answer the questions. And you need to be able to assess knowledge and understanding of the topic by completing a check test. So what is a respirometer? So a respirometer is a piece of apparatus used to measure the rate of respiration. So this is a picture of a respirometer. So you see an organism in the, so an insect in this test tube, so it's stuck in a gauze basket. And then you've got your delivery tube, and then you've got your manometer, so your U-tube collecting coloured liquid. And then on the other side, you've got nothing in the other test tube with your delivery tube. So you've got a potassium hydroxide, which is used to absorb carbon dioxide. So what would happen is, as this insect carried out respiration, your liquid would draw up. And the reason being is every time this insect carries out respiration, it's going to produce carbon dioxide. But the carbon dioxide is going to be absorbed by your potassium hydroxide, so it's going to create a sucking action like if you were sucking up straw. So every time the carbon dioxide is absorbed, the liquid is going to rise up. So you should then get an uneven scale, so it should be coming towards your insect because there's nothing in this chamber. So this chamber acts as a control. So it proves that the insect is having the effect. So you can go to this video which explains how you measure your rate of respiration by a respirometer. So you would want to make a note on your respirometer and your respiratory rate. So the respiratory rate is the rate at which an organism converts glucose to carbon dioxide and water. It can be calculated by measuring an organism's rate of oxygen consumption. So basically how much volume of oxygen the organism consumes. So studies on simple animals often use a respirometer. Respirometers measure the change in gas volume in a enclosed system. So any changes due to the respiratory activity of the organism that's being studied. So potassium hydroxide or soda lime is used to absorb the carbon dioxide produced, meaning that any change in volume are due to oxygen consumption. So this is a different type of respirometer this time, and it's measuring respiration with, well, in germinating sunflower seeds. So we've got our respiratory chamber, we've got a sodium hydroxide solution to absorb carbon dioxide with filter paper, and we've got a scale and coloured liquid to measure the uptake of oxygen. So our respirometers measure respiration as oxygen uptake per minute. So your carbon dioxide is released and is absorbed by sodium hydroxide, and that is how we measure our oxygen uptake because it creates sucking action. So cold-blooded animals are used as heat is released by warm-blooded animals, and that can affect the volume of gas in test tubes. And the temperature must be kept at a constant rate during the experiment by placing it in a water bath so it doesn't affect the volume of gas. And control tubes containing glass beads or non-respiring dead organisms must be set up so that it acts as a comparison. Remember, we've went through controls in various different video tutorials. So controls are always to act as a comparison to prove that whatever you're looking at is having the effect. So for this experiment, it would be to prove that the germinating sunflower seeds are having an effect. So you'd set up the exact same experiment, except from you'd replace the germinating peas with glass beads. And what would be expected is that the coloured dye would stay in exactly the same place, which would prove that your germinating sunflower seeds were having the effect and causing the oxygen uptake per minute. So peas are often used, but be careful not to assume that they are photosynthesizing. So for this respirometer, we have a snail, we've got our coloured liquid, and we've got our capillary tube, and we've got our solution to absorb our carbon dioxide. And where we get questions about this, and you can print out this is in activities and consumables. So our questions are, what causes the liquid level to change? And the second question is, what would be a suitable control for this experiment? So if you want to pause the video and then I'll give you the answers. So the answers to the questions are that as the snail respires, the snail will use up oxygen, and then obviously when it's using up oxygen, it will produce carbon dioxide. And then because the carbon dioxide is being absorbed by the sodium hydroxide or other substance, this causes the coloured liquid to move up the tube by a sucking action like a straw. And for a suitable control, you would replace the snail with glass beads of the same mass, or you would use a dead snail, but you'd keep everything else in the experiment the same. So it's important you put what you would replace it with and that you'd keep everything else in the experiment the same. So we have a problem solving exercise here. So respiration in germinating seeds. So the temperature at the start was 20 degrees Celsius. After 3 days, the temperature in flask A had risen by 3 degrees Celsius and flask B had stayed the same. So you've got your germinating seeds set up in A, and you've got your thermometer coming out, and you've got your boiled seeds set up in B. So what we're getting asked is what is causing the temperature to rise in A, and the second question is why was the dead flask of dead peas also set up. So if you want to pause the video and try to answer the questions, and then I'll reveal the answers. So the answers are that because the living peas are respiring in A, which gives off heat, so that would cause the temperature to rise, and the boiled peas were set up as a control because obviously the peas would not be alive. So past paper questions to review our knowledge on respiration. So it says explain why muscle cells require many mitochondria. And then for the second question, it says name one substance produced by a cell carrying out aerobic respiration. And then for B, it says a muscle cell will carry out fermentation when oxygen is not available. Describe the fermentation pathway in a muscle cell, and that is worth 3 marks. So if you want to pause the video and try to answer the questions, if you're struggling, you'll need to go back to the start of this video tutorial. So the answer to these questions are that your muscle cell requires a lot of energy, it's got a high energy demand, so it needs ATP for movement and contraction, so that's why it contains lots of mitochondria. For the second question, your products of aerobic respiration are carbon dioxide, water, and lots of ATP. You only needed to mention one of them. And for B, your glucose is converted and broken down to 2 molecules of pyruvate and at the same time produces 2 ATP in the cytoplasm, and then your pyruvate is converted to lactate. So you've got 1 mark for each of them stages. So well done if you got all of these questions correct. If you didn't, you need to go back to the beginning of the video. So then you're going to answer these questions in your jotter. So how is respiration controlled? Name the high energy compound produced in respiration. Give 2 things that energy produced in respiration can be used for. Where does aerobic respiration take place? What are the products of fermentation in animals, plants, and yeast cells? And what piece of equipment is used to measure the rate of respiration? So if you want to pause the video and answer the questions, and then I'll go over the answers. So the answers are: the respiration is controlled by enzymes, and the high energy compound produced is ATP. And ATP is used for muscle contraction, cell division, protein synthesis, and transmission of nerve impulses, and also active transport. And for where does aerobic respiration take place? It starts in the cytoplasm, so glycolysis happens in the cytoplasm, and it is completed in the mitochondria. You need both of the answers, or you won't get your mark. And then for 5, your end product in your animal cell for fermentation would be lactate. Your end product for fermentation in your plant and yeast cells would be ethanol and carbon dioxide. And 6, respiration is measured by a respirometer, which measures your rate of oxygen uptake per minute. So your tasks now are to review your knowledge of respiration. So you should complete cell biology exam practice questions, page 9 to 11, and you should find that in Teacher's Guide and Exam Practice book. And then you want to complete the respiration past paper questions, and that can be found in the folder 4, past papers, and mark it until you get 100%. Same with the respirometer past papers, which can be found in the same place. And then after you've done all of that and completed all that, you want to try to complete the respiration homework, and it's important that you send it to your teacher for marking so that we know that you're understanding the course, and so that you know that you're understanding the course. So well done if you've got this far. If you're struggling with respiration and you find some of the questions difficult, remember you can watch the video as many times as you need to. So we've got different uses of fermentation. This is more National 4 work than National 5, but it's useful to know anyway. So humans use the process of fermentation to make useful products, e.g. bread, beer, and wine. So we know that fermentation causes in yeast cells the breakdown of glucose into ethanol and carbon dioxide. And yeast is a single-celled fungi that feeds on glucose, and then obviously carbon dioxide bubbles, so it can make dough rise, and then the ethanol would evaporate in the oven. So in fermentation, we've got glucose, which produces your pyruvate and your 2 ATP, and then it produces ethanol and carbon dioxide, which can be used in the baking industry. It can also be used in the brewing industry, so the ingredients for beer can be added to large tanks called fermenters. So yeast changes glucose into ethanol by the process of fermentation, which we've already explained, and they get made in these giant fermenters, which are up in the corner. So they have computers which control the temperature and the pH to provide optimum conditions for yeast growing, so the best conditions basically for the yeast to grow. So fermentation and renewable energy. So biorenewables are types of renewable energy produced by living things. So examples of biorenewables are biofuel, biodiesel, and biogas. So biofuel, e.g. ethanol. Ethanol is made by fermenting the sugar in sugarcane, sugar beet, wheat, maize, or wheat pulp. Biodiesel, so e.g. alcohol and oils. So biodiesel is produced by mixing alcohol with oils that have been extracted from plants such as rapeseed, soybean, or from waste vegetable oils. And then biogas, e.g. methane. So biogas is produced from decomposing animal or plant waste. So then we have fermentation in dairy industries. So in dairy industries is yogurt making. So there's a sugar in your milk and your yogurt called lactose, and when your bacteria act on it, they convert it into lactic acid, which makes a sour taste. So bacteria feed on lactose sugar in milk, and the milk clots and becomes acidic, and that makes yogurt and gives it that taste, so like strawberry taste, etc. Then we've got fermentation in dairy industries again. So this time bacteria is used to make cheese. So bacteria feed on lactose sugar in milk. The milk clots and becomes acidic, making yogurt. The enzyme rennet is then added, and then rennet clots the proteins in milk to make curds, and the liquid part left is called whey. So the curds go together to form the cheese, and then the liquid whey is then filtered off. So to summarize fermentation, you would need to draw this table for in the case of microorganisms. So your type of microbe that would be used in brewing would be your yeast and fungi. So the important part would be your glucose is converted to ethanol, and then ethanol would be used to produce alcohol. For baking, it would also be your fungi yeast. So glucose is converted to carbon dioxide, and your carbon dioxide makes your dough rise. And then for the last one, yogurt. So your type of microbe would be your bacteria. So your lactose sugar is what your bacteria feed on, and they produce lactic acid, which produces the flavour in yogurt and in cheese. So well done if you finished this video. That is the end of cell biology. Woohoo! So basically that's all of the videos and work that you need to complete if you've completed all of your homeworks and various other textbook questions. That's everything that you need to do until you come back to school after the summer holiday. So very, very well done to you. You can give yourself a pat on the back. Remember, if you're struggling with anything, go back because you will be getting tested when we come back from the holidays, probably in the second week, maybe even the first week. So make sure you go over everything in great detail and make sure you know all of the different key areas well. So big, big well done to everybody and see you after the holidays.